So I welcome you all here to the orientation program. This is for our foundation classes. It is a very important bridge course where we are joining the secondary school students, that is those who are currently in class 9th and class 10th, with the upcoming sessions of 11, 12. So by the time these 2 years they study their foundation, they are already having a strong base for how to overcome those bulky syllabus, how to combat those things like how to deal with 11th and 12th sudden changes and rise in the level of syllabus and concepts. So BioAS has taken this initiative. So let's have a combined efforts of a foundation where there is a amalgamation of a syllabus of biology for class 11th and 10th both. And here I'm going to tell you about those chapters as well. So before we go for this, I would like to give you a brief idea that how we enter into the senior secondary wings. Also, they also undergo some kind of, you can say, even they also undergo participation in the orientation. So what we make them understand, same kind of thing we are going to make you also as well, right? So for the today's day, you all are supposed to, whatever you want to ask, your questions and queries, you feel free to write in the chat box. Once the session is going to take a pause, at that time I will look at your chats and will speak on that. So here it is about the orientation. So if we see, that is BioAce. If we talk about myself, I'm Varsha Roy. I'm the founder and CEO of BioAce, looking into this line for the last 18 years. But in Faridabad, I have given 11 batches of NEET, and that too with the highest score of 360 out of 360 in biology for the last consecutive 11 years. And here in our institute, it is the 5th year running. And here also we have the highest of 355 in biology, and 85% of the students are scoring 300 plus in biology in the NEET NTA exams. I'm an author as well. I have a special bent of mind to write a lot on NEET books, latest programs, new type of learning tricks, which is very much good for the students. So it's completely a curated, you can say, the platform for the students' requirement. So here, so all of us are here together to ensure that if there is something, a beneficial thing, product in the market, why not the student should also be a part of it? So recording. So what happens is, in this case, we have there is a like this. So what we are providing you guys, this foundation course. So this foundation course is very uh, important platform for you all, right? Next, we have our strength. Though it is for 11th and 12th, I'm introducing you just because so that you know in the future what we have and you can tell others as well. That is, we have 2 doctorates. They are the very good scientists from BHU, Banaras University, and the University of Delhi. So both of them are very good writers as well as they are very good professors and they teach very well. We are having them in our panel so that we can give the students a very good depth of the foundation and knowledge, so that there is any kind of doubt sessions or something, then they will be the right person to help you out. So this is all comes in this way, that how the BioAce is working day and night for making new programs exclusively for those students who are genuinely want to study biology as a career, or at least wherever any competitions, whether it is non-med, whether it is commerce students ka bank PO services, whether there is a biology in the course, we are there to help them out. After this, we also have this search. We have this is app-based education with us, which is curated only for the BioAce students exclusively. And the students gets the credentials. They are able to enter into this. They get everything related to the foundations. And entire year, they have lots of quiz, test series, everything with the certification. So it is also supporting Android as well as iOS software. Then we have, it's about me, like I'm also in the panel of Olympiad, designing of question papers, like for humanity Olympiads. And then various teacher training programs. I go to various places. in Lucknow, Coimbatore as well. I was also for a long time into the panel of Jaipur India groups for teacher trainers. And then this is all about the chapters. Now here, if I skip the chapters part of 11th, I will give you a mixed chapters of 9th and 10th. I'm coming to that also. You can note it down what all chapters we are going to do. It is a course of 10 months. So within 10 months, what all best I will going to give you, I will provide you that, right? So it is starting from April to December end, because January you have your own in-house exam starting. So we set you free from there. So we have the biology results. These are exclusively bio-based results. Then even the academic results of pandemic was very good, and we have the results also in front of you. All the range we have mentioned, all the grade of range we have mentioned. We don't hide anything. That whatever we have students are getting, we have shown it. So and our this year's placements to various government medical colleges are also shown here, right? So this is how we work together. We have a very strong alumni team, and today also we have invited our alumni from an MBBS college, and he's going to speak on us that how we were helpful for him to the class 12th and outgoing students. So this is the moment with the students. We make it very good, and we have an annual day celebration. We also have seminars and webinars where the various counselors for admissions in India and abroad as well. And this is how best of luck for all those who really want to do very well in our education system. And thank you for giving your time to BioAce, right? So now I'm going to tell you about the chapters which we are going to study. So the chapters, please note it down for everyone that what all chapters are important. It has been taken into consideration the weightage in the future. So the chapters are, it is a combined integrated, it is integrated foundation. So it is also covering 9th and 10th board. So it is integrated foundation course, and chapters which we are going to deal are, please note it, first cell, fundamental unit of life, fundamental unit of life. Second, we will be having tissues also. Tissues, both plants and animals will be considered. So we are discussing intact on all living organisms. In all living organisms, whatever the type of tissue, please note it down. We are going to teach you, right? And after this, the third chapter will be diversity, but we will deal it later. First, we will talk about life processes. Life processes in which we will discuss about nutrition, respiration, transportation, and excretion. We will also talk about, after this, reproduction in living organisms. Fifth, we have, after this diversity chapter, we will be discussing the diversity of living world, diversity of living world. This is a specially curated package which I have decided for the students and what is best for them, looking into the necessities. And then we will talk about genetics and biotechnology, a brief introduction. A brief introduction. And then so cell, tissues, life process, reproduction, diversity, genetics and biotechnology we have. And after this, next chapter is going to be our control and coordination. So we talk about control and control and coordination. Right? These are the chapters. Right? Control and coordination. So like this, we will be discussing about the topics. And of course, how can we ignore the eighth topic, and that is environment related basic. We can say fundamental ecology. Let's name it fundamental ecology. Because in any stream, you should know about the environment, the type of petitions filed, and the type of, we can say, any things, any news happening around, and then any kind of new changes in the environment. What are the new laws? So any kind of like right now is G20 going on. So what all the protocols have been following for the ecology benefits of to mitigate the pollution. So we are going to discuss all that current affairs kind of fundamental ecology में. तो ये सारे हमारे 8 चैप्टर्स का बैकअप रहेगा. हम ये पढ़ेंगे. मैंने स्पेशली आप लोगों के लिए डिजाइन किया है कि आप 9th में है या 10th स्टूडेंट है, आपको ये बताना चाहिए, आपको ये आना चाहिए. So this is very important. So please note it down. So this is the foundation series for NEET NTSC Olympiads, and let's talk about them. So here the modules are there. So first of all, let's talk about a very important module that is starting from the cell, the fundamental unit of life. So how you define cell? Fundamental unit of life. First, let's have a theory. What do you know about the cells? Cell is structural and functional of life forms. No need to write them, they're already with you. जहाँ भी लगेगा आपको लिखना है, आप वही लिखेंगे, right? So first of all, you make this flow chart. That cell is having division structure-wise, shape of the cells, number and size of cells, types of cells. So we will study first of all the various components. So let's study about shape and numbers. We will study. Then we will talk about the types. Types with prokaryotic, eukaryotic, plant cells, animal cells are there. Right? And what are the living parts of the cell? Cell membrane, nucleus, and cytoplasm. What are the structure of the cells? Cell membranes, nucleus, cytoplasm. Cytoplasm means cell organelles are present. So like this, we are going to study about the cell. So like the notes, please. Whatever I will tell you, note that part only. So here you have to write, note it down from here till here. Flow chart. Okay. So here after this, we see that cell is the structural and functional unit of all life forms. You all agree? No need to write this, we study from very beginning. Now single cell organisms, top one है. Bacteria, amoeba, chemadomonas, all are having single life, they all are showing the same life processes inside them. Microscopic study of cell is called cytology. So write this point, the microscopic study is known as cytology. So what is the definition of a cell? It is a unit of protoplasm surrounded by a membrane. A unit of protoplasm surrounded by a membrane. Note this. Next we'll talk about the history. History में Leeuwenhoek, the first person to construct a microscope. You have not studied about this already, right? So it was the Leeuwenhoek, the first person to construct a microscope. He discovered the minute forms of life such as bacteria, single cell animals like infusoria in a drop of water. Yeah, single cell animals, infusoria in a drop of water. Cells were first discovered by Robert Hooke in 1665. Everyone knows about it. And he also wrote about it. He studied the cork slice. He wrote a book, Micrographia. This is a question. Whatever I'm teaching you is all questions and Olympiads. Right? So it is a honeycomb-like lattice-like structure. Honeycomb lattice structure. Then Leeuwenhoek, in 74, with improved microscope, he discovered the free cells of bacteria for the first time. So who saw it for the first time? Again, Leeuwenhoek in better improved microscope. He was able to see a more minute organism, bacteria. Robert Brown, you must have heard this also, but you can write it. 1831 discovered nucleus. Purkinje in 1839, he was the one to term the protoplasm, that is the living fluid substance of the cell. So how these questions comes? There is a trick in my foundations. You will always have a this Q. If I add a Q means it is very important, has been asked several times. It's a PYQ. What is PYQ? Previous year questions. लिखो ये सारे applications. ये आप सबके लिए नया है. लिखो. एक index बनाओ. Index of shortcuts. In my ways, PYQ. Previous year questions. If I write essay, it means it is fill in the blanks. Start writing. If I write MTC, it is match the column. If I write GR, it is asked as give reason. And if the bracket, I have bracketed any sentence, then I'm writing A or B oblique as in circle. It means it is assertion reason. It is assertion reason. Right. CF, that means true and false. It means statement questions. These days this is trending. In these years, this is the most trending statement questions. So now the foundation class will start this at the beginning so that your 11th and 12th is going to rock. Right? So please note this down. And of course, the last also, who proposed the cell theory? The biologist, Sheldon and Schwann. But Sheldon is a botanist, Schwann was a zoologist. Rudolf Virchow was the one to write omnis cellula e cellulae, that is all cell arise from pre-existing cells. So this is the important history. History पे questions कैसे आएंगे? History पे questions आते हैं as match the column. Who did what? Scientist name in column 1 and in column 2 what they did. So note it down. Okay, note it. Next, we have after the history, we have the fundamentals in cell theory. What are the cell theory? Don't need to write this in general. That it was postulated by all there's Sheldon, Schwann, and then later by Rudolf Virchow. All plants and animals are composed of cells. Means all living organisms are composed of cells. Cell is the basic unit of life, and all cells arise from pre-existing cells. So, of course, we should know who is the exception to the cell theory. Viruses are exceptions of cell theory. All right. Types of cells and organisms. So, what are the various types of cells and organisms? On the basis of number of cells, organisms can be as unicellular or multicellular organisms. Okay. So, who will show division of labor? Unicellular or a multicellular? Multicellular. So, please note it here that it is showing division of labor. Showing division of labor and is more complex. And is more complex. It's just showing division of labor and is more complex. Done here, unicellular and multicellular organisms, right? So please write down the major difference, write down on the basis of. Acha, always remember if I write my notes on the basis of OBO, I write always OBO on the basis of number of cells organisms can be categorized as. So unicellular or multicellular. So note it down, and then on the basis of organization of nucleus, write down organization of nucleus. So nucleus, prokaryotes and eukaryotes. So prokaryotic cells, eukaryotic cells. So who is primitive? Pro means primitive. Karyon means nucleus. So it is primitive nucleus. Similarly, U means advanced. Advanced, and karyon means nucleus. So prokaryotic and eukaryotic cells. So these are primitive. These are primitive. They have no cell organelles, double membrane वाले bounded organelles नहीं है. They don't have proper nuclear membrane. They don't have nuclear membrane and nucleolus is absent. This is जब भी मैं encircle करके minus लिखूं इसका मतलब absent और जब भी plus लगाऊं तो present. So eukaryotic cells, they are well developed. They have advanced nucleus. Advanced nucleus. So like this, we have the organisms. Next, cell structure, shape, size, and number. So in this, we will see structure of cell, structure, shape, size, and number. Structure of cell. All cells have three major functional regions: plasma membrane, nucleus, cytoplasm. You note here, something, right? Now, the cytologists divide the cell into two major components: the nucleus and the cytoplasm within the development of electron microscope. So, what is the magnification order of electron microscope? Note it down. This one, 16 into 10 to the power 5 is the magnification of electron microscope. Shape of cells, there is a great variation in different shapes of the cells, right? And it is related to its function. Very important. It is related to its function. So it can be asked as standalone MCQ. So note it down. That shape of a cell, there is a great variation in the shapes of cells, and the shape of cell is usually related to its function, right? Then, after this, here, can you identify? So this can come as an exam, maybe identify the, what they can be asked, identify the differentiated cells, differentiated TEA, differentiated, differentiated cells. So first of all, this is smooth muscle, spindle shape. लिखो इसको draw करो. Draw this spindle shaped. This one is biconcave. क्योंकि इसी की वजह ये आपके उसका आ जाएगा ना in your exams. Which type of cell has which type of shape? So blood cells biconcave, enucleated. अच्छा अगर वो RBC है तो enucleated है. अब ये WBC है. This is a WBC. Then nerve cell, it is elongated with arborization. So what is arborization? Means branching. Head पे भी, tail पे भी. There are dendrites and there is nerve endings. So this is arborization, means branching. So it is identify the differentiated cells which is elongated with arborization. Fat cells, here this is the adipose. It is also known as adipocytes. Adipocytes, where this is a nucleus at the periphery, ring-like structure. लिखो इसमें signet, ring-like structure. Signet, ring-like structure. It means that there is a huge fat globule. This is a fat globule inside which fat is there and it is shifting the nucleus to the periphery. Then we talk about the bone cells. Bone cells is also known as osteocyte. Write out osteocyte. Ovum, the egg, and the sperm which is flagellated. Flagellated for mobility. It can move for mobility. For mobility. So note it down. This also you can get from your books, not a big deal. Inside of the cells, the smallest cell कितना है 0.12 0.2 micrometer are found as bacteria. And what is the name of this? PPLO. So what is the full form of this? Yes, so remember this will be a question. The smallest cell, Mycoplasma family. It belongs to Mycoplasma, right? So it is PPLO, pleuropneumonia-like organisms. Mycoplasma. इसी को बोल देते थे और करके लिख देते थे Mycoplasma. ये रहा एग्जाम्पल है ना Mycoplasma jelly-septium. So note it down. The largest cell is the egg of an ostrich. Egg of an ostrich, and what is the dimension? 180 millimeter in diameter. Largest human cell is a female ovum, and smallest human cell is RBCs. Then longest कौन है? Nerve cell. The only flagellated cell कौन है? Human body में sperm cell. Sperm cells, which is motile, right? So like this is about the humans. So you should remember them. Now cells are measured in micrometer. There are units. This is the unit of microscopy. A micrometer is 1000 of millimeter. So micrometer is equal to 10 to the power -6 meters. And if it is nanometer, 10 to the power -9 meters. Right? So like this we have. So this is about the measurements. Number of cells. Number of cells in living organisms also varies greatly. So number of cells भी vary करेगा as per the complexity of the organism. Unicellular is made up of single cell, like Chlamydomonas, amoeba, paramecium, protista organisms. And in the human body also we have trillions of cells, 26 trillion cells. So होने में कोई एक सेल का बने कुछ few thousands के बने हुए कुछ few hundred cells के बने हुए और बनने में ऐसे trillion के बने हुए. So this is a range number of cells. Right? Please note it. A microscope is an instrument in microbiology we are using it, which magnifies the object to a particular range, right? So every lens has its own power. Accordingly, it is able to magnify the objects. So resolving power of a microscope varies. In addition to magnifying a specimen, what we have to do is it should be understood by the closely spaced parts. The ability to provide such an image is called resolving power of a microscope. What is resolving power? Microscope must produce a clear image of the closely spaced parts. So जो भी बहुत ही पास में कंडेंस सेल ऑर्गेनल्स हैं उसको इजीली दिखा सके अच्छे से. इसके लिए स्टेनिंग प्रोसेस भी चाहिए so that we can able to see them nicely. This is microscope, a simple compound microscope of the labs. There is an eyepiece जहाँ से observer देखते हैं. Force adjustment, fine adjustment, blurriness को हटाने के लिए. And this is the stage where the objective lens is placed. कौन से power आपको लगानी है यहाँ लगाएंगे. माइक्रोस्कोप की स्लाइड यहाँ रख देंगे उसकी क्लिपिंग कर देंगे ताकि वो अपनी जगह से हिले नहीं एडजस्ट करने के लिए करते टाइम. स्टेज है ये पूरा स्टेज जिस पे वो रखा जाएगा. स्लाइड ग्लास स्लाइड विद कवर स्लिप. कंडेंसर है टू फोकस द लाइट ऑन द क्लिप ओनली माइक्रोस्कोप स्लाइड पर विद द मिरर. एंड सो दिस इज हाउ एंड द बेस as a stand to support on the ground. तो इस तरह से माइक्रोस्कोप रेडी होता है. हाँ, civil is a screwed parts. This part is attached with the stage. No, it's only coming for drawing. parts of a cell अगर हम देखें, so what are the various parts of a cell? Plasma membrane or cell membrane. Nucleus has bonding subparts. It is nuclear membrane, nucleolus, nucleoplasm, chromatin network. Cytoplasm contains various cell organelles like endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, plastids में plants में होता है only, ribosomes and centrosomes animal cells में होते हैं only in the animal cells. So you wrote down the parts of the cells, please. Wrote it. Next. Yes, so plasma membrane or the cell membrane. First of all, there are two names: plasma membrane, cell membrane, plasma lemma. सब एक ही बात है, right? So even in some organisms like your organisms, animal world, it is also known as white line membrane. That is also same thing. Egg की membrane, especially white line membrane word use कर देते हैं, but that is also a plasma membrane. So every living cell is extremely externally covered by a very thin transparent elastic semi permeable. It is selectively semi permeable. तो यहाँ पर एक question आ जाता है, what type of permeability is seen here? So is it only permeable, impermeable, selectively permeable? How is it? So you will opt for selectively permeable membrane. Okay, it is present in both type of cells, prokaryotes as well as eukaryotes. And what is it is made up of? Protein and phospholipids. So protein and what is the best studied model? Fluid mosaic model of plasma membrane is best studied, which is most acceptable, given by the scientists Singer and Nicholson. And it is having exchange of gases. Diffusion is accepting. It is accepting some specific type of movements from a region of higher concentration to the region of lower concentration. So there is a transport across the membrane. This is very important concept because of this only you are able to understand some basic principles which are taking place in class 11th as well. Of the plasma membrane, right? Diffusion, osmosis, and you also talk about active transport. So these are passive transport or active transport. So active transport is one where you need energy, ATP consumption, and passive transport is the one where you don't need the ATP or the any work. It is through concentration gradient. So that is we have to study here. All right. So please note down these points about the plasma membrane. Then we will switch on to the next, that is fluid mosaic model. We'll study fluid mosaic model. So the questions are there. And plant transport also. There are examples like osmosis. For example, you see here fragrance in a room, perfume हमने एक quantity sprinkle किया वो पूरे room में spread हो जाएगा. वो air के through diffusion हो रहा है. Now if a drop of ink is added to a beaker containing a water, further पूरा ink पानी में dissolve हो जाएगा. So that is because of diffusion. Liquid में भी diffusion है and solid phase में भी diffusion. Sorry, liquid में भी और gaseous. It is in fluids which is possible. So movement of water takes place for the process of osmosis fluid. Osmosis is also there. Movement of osmosis should be through a semi-permeable membrane. So what is osmosis? यहाँ पे diffusion word आएगा. यहाँ पे क्या आएगा? The movement of water takes place by the process of osmosis, but then we'll study in osmosis वाला paragraph, right? So it is a process of movement of solvent only. Who is in this solvent? Remember, it is the solvent molecules from the region of lower concentration or higher concentration. Yes, from the region of higher. Movement of molecules from the solvent, the region of I think it should be from this. Higher to lower concentration through a semi-permeable membrane. To a semi-permeable membrane, right? So, if a plant cell is placed in a concentrated solution, then the cytoplasm of the cell shrinks, right? And the cytoplasm shrinks, the water is universal. From the cytoplasm moves into the surrounding solution through cellular membrane. So this is there. So phenomenon is called osmosis. and the cell which has lost the water is known as flaccid cell. It is flaccid cells. Next, whenever you have to write, I will tell you. Like this one. So you can see here what is a condition. So normal plant cells. This is a normal plant cell is there. Suppose a normal plant cell plus concentrated solution is there. So osmosis में exosmosis हो रहा है. So the flaccid cell plus water, right? Exosmosis means loss of water. The cell is now flaccid with water out, and if this water is taken again back, so then it is a turgid cell. So there are tonic solutions. How is it happening? It is all because of tonic solutions. So whenever there is a water medium in which we have solutes, then the solvent जो कि जिसमें डिफ्यूजन इसमें वो पूरा है मिक्सिंग और ये है माइनर क्वांटिटी के सॉल्यूट्स. तो सॉल्यूट प्लस सॉल्वेंट क्या बनाता है? सॉल्यूशन. So depending on the concentration of solute, depending on the concentration of the solute, remember this. With respect to solute, with respect to solute concentration, solute concentration, with respect to solute concentration, what will happen? In this, if the solute concentration is very high, अगर solute का concentration बहुत ही high है, like this, then it is hypertonic with respect to somebody which is less solute. ठीक है? एक हमेशा with respect to होता है, तो इसके साथ एक और membrane लग जाएगी, इसके कम solute है, और ये पानी है, इसमें दो ही है ऐसे तो. This is hypotonic with the surrounding medium. और अगर दोनों फेज में सेमीपर्मेबल मेम्ब्रेन के दोनों तरफ अगर बराबर सॉल्यूट्स हैं, then it is isotonic solution. So hypertonic and hypotonic solutions are there, which we study. From our class? All right. So like this, we study about the solutions. Okay, so have you noted this sequence? So the normal plant cell plus concentrated solution to osmosis, exosmosis, flaccid हो जाएगा और ये. So it's a reversible process. So reversible process है, so it is a physical change, a kind of physical change. It is a physical change, right? In the plasma membrane, it is semi-permeable membrane. So now it is composed of lipid and protein bilayer also. So what is so special about it? These plasma membrane has two lipids layer. Can you see this? The two lipid layer, this one and this one. In this lipid layer, can you see the structure? It looks like a head, like this, and two tail-like structures. Looks like this. So exactly, this is hydrophilic end, means water loving, attracting. और ये वाला जो है long hydrocarbon chain, this is hydrophobic. Hydrophobic. This is a very important concept. इतने सारे medicines, research, R&D इसके व्यक्ति में बहुत ज्यादा है. क्योंकि ultimately सबको medication या pharmacology में प्लाज्मा मेम्ब्रेन के अंदर किसी को भी पहुंचाना है. अगर जब तक सेल के अंदर कोई नहीं जाएगा तो मेटाबॉलिज्म में इफेक्ट कैसे आएगा? इसलिए सारा जो गेम है वो पूरा प्लाज्मा मेम्ब्रेन के नेचर के ऊपर है. प्लाज्मा मेम्ब्रेन के नेचर को समझकर इसमें हम एक्सपेरिमेंट्स कर रहे हैं, साइंटिस्ट करते हैं. यहाँ पर ये है हाइड्रोफिलिक वाला जो रीजन है, ये वाला है, ये हाइड्रोफिलिक रीजन है, और ये पूरा हाइड्रोफोबिक रीजन है, जिसकी वजह से क्या बन जाता है, ये पूरा एक यहाँ से लेके यहाँ तक इट इज आइलैंड ऑफ हाइड्रोफोबिक, आइलैंड ऑफ हाइड्रोफोबिक मीडियम। दैट्स व्हाई लिपिड्स तो बहुत आसानी से, सपोज ये लिपिड है तो लिपिड्स बहुत आसानी से इसको क्रॉस कर जाते हैं और आराम से ये इसको स्पेस देकर उस पर से निकल जाते हैं। बट अगर ये वाटर है, अगर ये वाटर बॉर्न है, मतलब लिक्विड, मतलब इट इज रिलेटेड टू सम लिक्विड ऐसे, तो यहां पर ये इसको रिपेल करेगा, इसलिए इसके पास पोर्स या चैनल्स है। दैट्स द रिक्वायरमेंट ऑफ ट्रांसमेम्ब्रेन प्रोटीन। इंटीग्रल प्रोटीन की रिक्वायरमेंट इसलिए आती है बिकॉज जितने भी वाटर रिपेलर्स हैं, मोस्टली वाटर सॉल्वेंट, वाटर तो इसका सब सॉल्वेंट है, सबसे तो उसके पास कैटेगरी है। तो ट्रांसमेम्ब्रेन प्रोटीन्स ध्यान रखना इस चीज का कि जितने भी हाइड्रोफिलिक वाले हैं जो नहीं जा सकते डायरेक्टली थ्रू इस आइलैंड को क्रॉस नहीं कर सकते, लिपिड आइलैंड को, वो इस वे में जाएंगे, वो चैनल्स खोलेंगे पोर्स के लिए। तो पोरेंस के थ्रू, क्या नाम है इसका, पोरेंस, क्या बोलते हैं, पोरेंस, एक्वा चैनल्स होते हैं पोरेंस। देयर आर डिफरेंट डिफरेंट टाइप्स ऑफ पोरेंस, मतलब प्रोटीन structures, they are present in bits and patterns so that जहां पे जिसका गेट है easily वो वहां से उसका molecular size allow कर दे उसको जाने के लिए। That's why, तो ये होता है इसका रीजन। कोलेस्ट्रॉल का क्या फंक्शन आएगा, कोलेस्ट्रॉल की ये एक बेनिफिट मिल जाता है कि जो कोलेस्ट्रॉल है, दिस कोलेस्ट्रॉल इट इज इंश्योरिंग दैट कि ये जो फ्लूइडिटी है ये फ्लूइड मेंटेन करे, मेंटेन्स fluidity of the membrane. It maintains fluidity of the membrane. तो इससे क्या होगा, जो फ्लूइड है, मतलब जो कोलेस्ट्रॉल जो है ये आसानी से स्लाइड, मतलब ये जो है ओपन और क्लोज हो सकते हैं, ए वेरी फ्रीली मूवेबल, दे कैन मूव इन zigzag ऐसे और ट्रांसफॉर्म पैटर्न, क्रॉस पैटर्न से मूव कर सकते हैं, लेटरल मूवमेंट शो करते हैं, जिसकी वजह से क्या होगा, इसकी वजह से व्हाट हैपेंस, सो ड्यू टू दीस मूवमेंट्स, inter movements, they are able to create space for incoming and outgoing products, so that is there. Then what is the role of these glycolipids, these glycolipids and carbohydrates? Carbohydrates are mostly acting as antigen for recognition, cell to cell recognition. This is cell to cell recognition, cell to cell recognition, बाइंडिंग के लिए, मतलब एक तरह से बायोमेट्रिक एक्सेस बना के रखना है सेल के, कि ये उसके लिए जरूरी है कि एंट्री कराया जाए। सो ग्लाइकोलिपिड्स, अब ग्लाइकोलिपिड्स में, ग्लाइकोलिपिड्स आर आल्सो हेल्पिंग देम फॉर द सेम, फॉर मेम्ब्रेन बाइंडिंग। मेम्ब्रेन को मजबूत रखता है, बाइंडेड रखता है, उसको बिल्कुल अच्छे से उसका कॉन्स्टिट्यूएंट मेंटेन रखता है। सो दीज आर द वर्क ऑफ देम, राइट। साइटोस्केलेटन नीचे दिया हुआ है, ऐसे आप देख सकते हैं, लॉन्ग लॉन्ग ऐसे रॉड लाइक स्ट्रक्चर्स हैं साइटोस्केलेटन, दे आर फॉर सपोर्ट, दे आर फॉर सपोर्ट एज अ बेस, एंड देन यहां से इनसाइड साइटोप्लाज्म शुरू होता है। तो ये प्लाज्मा मेम्ब्रेन का इनसाइड है, facing the cytoplasm, again a water medium, and outside is also extracellular space, it is also following the, this is also following the extracellular space, this is also following the, facing the water medium, ECF, extracellular मतलब सेल के बाहर, extra word in zoology means outside to something, तो extracellular सेल के बाहर वाली, right? So like this we can see here that this is about the plasma membrane and its constituents with its functions. ये बहुत इंपॉर्टेंट फीचर्स हैं, आपको लिखना होगा कि इसमें कौन कौन से चैनल्स वगैरह प्रेजेंट है, क्या क्या इसके फंक्शंस है। सो फंक्शंस में अगर हम देखें तो डिफ्यूजन इज एबल टू टेक प्लेस वेरी इजीली बिकॉज हायर कंसंट्रेशन टू लोअर कंसंट्रेशन। डिफ्यूजन हो गया। डिफ्यूजन में भी अगर ये पैसिव में आएगा, this comes in passive. Let me change the screen page. I'll give you a fresh note, right? Yes. So here, transport across cell membrane. So in this transport across cell membrane, what happens? In the transport across cell membrane, what happens? First of all, what will happen? How will it happen? First of all, it is an passive transport. It is passive transport, and here it is active transport. Passive and active transport. Passive transport may be it is osmosis, diffusion, ठीक है, और एक होता है facilitated diffusion. Facilitated diffusion. Facilitated diffusion. In this ATP cross carrier protein porins, जिसे हम porins भी कहते हैं, क्रॉस टिक इसमें चाहिए, और एक्टिव में एटीपी टिक। अच्छा, कॉन्सेंट्रेशन ग्रेडियंट मतलब अगेन इज इट फ्रॉम लो टू हाई और हाई टू लो? सो इट इज हाई टू लो टुवर्ड्स कॉन्सेंट्रेशन ग्रेडियंट, दोनों ही टुवर्ड्स कॉन्सेंट्रेशन ग्रेडियंट, और ये है एक्टिव against concentration gradient. Against concentration gradient का मतलब होगा that it is moving from lower concentration to higher concentration. Example best studied in your biology is sodium potassium acid. Sodium potassium ATPase pump. Sodium potassium ATPase pump. So passive active transfer across cell membranes are of these kinds. So we should know three criteria: ATP is used or not, carrier protein is used or not, or concentration gradient level. So on this basis, transport is taking place. So active transport is very much in demand. So there is a lot of things where active transport take place. So sodium potassium ATPase pump is the most common one, which is consuming your more than 70% of the energy is consumed here in the neural tissues or the neural tissues. And passive osmosis taking place for the water wherever the water is involved. Diffusion wherever the gases are involved. Like respiration, respiratory membranes. So what would be the example for diffusion? Respiratory membrane at the alveoli or blood. So respiratory membranes पर ये हो रहा है at the respiratory membranes. और अगर हम osmosis की बात करें तो it is for the water that is taking place in the gut walls, gut epithelium, for water absorption areas. Yes. Difference ये है कि उसको protein carrier चाहिए. There is a requirement of protein carrier in facilitated diffusion. Means ये membrane है, ठीक है, और यहाँ पे carrier protein है, यहाँ पर एक substance आके उससे जुड़ गया और ये ऐसे इस तरह से घूम गया 180 पर, तो ये carrier protein अब किस तरफ चला गया अपने आप इस तरफ, तो ये इसको बाहर निकाल देगा, उसको बाइंड आउट कर, उसको उसकी बाइंडिंग खुल जाएगी, वो से डिटैच हो जाएगा, ये वापस ऐसे करके लूप करके वापस यहाँ फेस करेगा. इस तरह से ये इन एंड आउट करता रहता है, मतलब सेल का एक हिस्सा पकड़ता है और उसको दूसरी तरफ छोड़ देता है. वो तो डायरेक्ट पास कर जाते हैं विदाउट एनी कैरियर प्रोबलेम्स इसको बोलते हैं फैसिलिटेटेड डिफ्यूजन so like this we have membrane transport all right so अभी अभी यहाँ तक किसी को कुछ भी पूछना है तो ask